We are in the last session of human reproduction, that is male reproductive system. In the last session, we have discussed regarding ductal system of a male. In this particular session, we are discussing regarding accessory reproductive glands. We have already discussed a little bit of them. Here, the first will be seminal vesicle, which is a sac-like structure which is always present in a pair. I'll show you in the diagram. They are present just near to the urinary bladder, as you can see here the labels, right? They are in pair, they are a sac-like structure and they'll produce fluid, namely seminal plasma. You can see that both the vas deferences which are coming on the external surface from the testes are merging after they'll be having the secretion of seminal plasma into it. And those vas deferens will then fuse with the urethra, which is coming from urinary bladder, and form the ejaculatory duct or viper duct, as the label is seeing in this particular diagram, right? Okay, so after all, the urethra is then having the secretion of prostate gland and the following bulbourethral gland. You can see the largest structure, which is single, the bulgy part as prostate gland, and the smallest P-shaped glands on both the sides of urethra, that is bulbourethral gland. So I'll be just briefly reading the content of those glands now. Fine. So seminal vesicle are present in pair. They're situated just at the base of urinary bladder. They'll secrete seminal fluid, or we have already discussed seminal plasma, which is the main content of the semen. That means seminal plasma is basically 70% and the rest 30% has been made from the secretion of prostate gland as well as bulbourethral gland, so that the semen what is given to the sperm as well as the fluid which is containing sperm. Fine. Okay, so seminal fluid is very viscous, it is alkaline, it is rich in fructose, citrate, prostaglandin and various kinds of protein to give detailed or proper nourishment to the sperms. The next one is a single gland, which is prostate gland, which is a single large gland, and that will produce milky alkaline secretion again to neutralize the acidity present in the vagina during the intercourse or sex, to avail the entry of sperm into vagina. The smooth and easy passage of sperms into the vaginal tract has been aided by this particular gland by neutralizing the acidity, so that motility of sperm can be maintained at pH nearly 6 to 6.5. Then the bulbourethral gland, which is also termed as Copper's gland, which is present in pair on the either sides of urethra, which is also secreting alkaline mucus fluid into the penile urethra, as we have seen in the diagram that is just present above the penis. So it is directly pouring its content into the penile urethra to aid lubrication or to provide friction-free movement of the sperm outside the body through the penis. Fine. Okay, so now we are discussing about semen. Semen is what? Basically sperm plus the content or the fluid which has been provided by these three glands. So the general name to the fluid which is produced by these three glands is given as seminal plasma, as we have seen earlier, which is nearly 95%, and the rest material 95, you can say 90 to 95%, as the sperm content is 8 to 10% in the semen, that is nearly 200 to 300 millions. So those numbers of sperms are ejaculated per discharge or per ejection in 3 to 5 ml of the content of semen, and the pH of that is 7.35 to nearly 7.5. The next thing we need to discuss in this session is spermatogenesis, that we have already discussed while discussing seminiferous tubule in second session, I guess, or third session maybe. This particular diagram is showing the cross section of seminiferous tubule, in which the spermatogonium are lined. They are the main cells which are undergoing meiosis to produce spermatozoas, that is the healthy and mature sperm. First, the spermatogonial cell will do mitosis, then it will do meiosis 1, as you can see in this particular diagram, to reduce the number of chromosome as half. Those cells are then considered as secondary spermatocytes, and finally they'll do secondary meiosis to produce spermatids which are round in shape. They'll then undergo the cell differentiation process to give the cell or a sperm a particular shape for its particular function. So the process is termed as cell differentiation to produce mature sperm cells. They can also be said spermatozoas. So there are three terms that you need to understand. The overall process of spermatogenesis is the sperm synthesis process, which has been detailed here in the two diagrams. The second important terminology is spermiogenesis, which is the last step. As you can see here, spermatids are converted into mature sperm cells in a specific shape. That differentiation process is termed as spermiogenesis. And the last step which is occurring here is spermiation, that is the release of mature sperm from this particular seminiferous tubule towards the ductal system of the male. Fine. After having nourishment from the Sertoli cell. Okay. So these are the three very important terminology that you need to focus. Now, the last part of this particular session is a structure of sperm. I'll show you the diagram of the sperm, which is having a head, the neck portion, the middle piece, and the last portion is a flagellum or tail. Fine. Now the head portion is basically having acrosome and a haploid nuclei. That acrosome content which is present at the tip of the head of the sperm is basically having the digestive enzymes called lyases and hyaluronidase, which has ability to penetrate the wall, or specifically termed as the membrane of the ovum. Fine. The next portion which is labeled as centrioles, which are mainly responsible for production of microtubules, which will give a particular shape to the sperm. The next is the neck portion, which is giving rotating ability of the head to the overall sperm. The next is middle piece, which are having numbers of mitochondria inside to provide movement of the particular sperm, or you can say they will avail motility to the sperm. They'll provide energy for the motility. That is also you can say. Fine. And the last portion is the tail or flagellum. You can say that by the research it has been identified that if the tail or a flagellum is rotating in a clockwise manner, the particular cell will go in forward direction, and if the tail is rotating in the anti-clockwise direction, the particular cell is coming backward. So by using flagellum, the sperm can move ahead or backward as per the need. Okay. So these are some of the important information or the final information regarding male reproductive system. If you like the video, kindly subscribe. Inside Biology by Dr. Pooja Brijesh. Thank you so much, guys.